The next company to present is iCoat Medical, and I am pleased to introduce CEO Peder Van and Head of Strategy David Berglund. Before I hand over the word to Peder and David, I can confirm as a point of information that iCoat will be carrying out a pre-IPO private placement. Over to you, Peder.
Thank you very much, Aron. Good afternoon, everyone. Thank you for being here with us today. My name is Peter. I am CEO and one of the co-founders of iCoat, and I have with me my colleague David Berglund, our head of strategy. If we move forward, please, 2 slides, I believe. Good. So I am one of the few ones in our firm who's not an MD or a PhD or a professor within immunology. Instead, I have a general background in business development, strategy consulting, as well as building an industrial compounder known as Diptech. But I can truly say that nothing has been more exciting than the journey we're doing with iCoat. So I'm very pleased to be here today and present it together with David. I'm going to walk you through 3 main areas today. First, I would like to give a brief introduction to our company, then demonstrate the medical need we are addressing, as well as the preclinical package upon we are building iCoat, and finally what lies ahead. Like Adam from ABG said, we are currently conducting a pre-IPO process together with ABG, and our aim is to raise money now before heading towards an IPO on First North in Stockholm, Sweden within 18 months. So if we move forward to the next slide, please. Now, iCoat Medical as a company is a fairly young one, but it is built around and upon a very, very strong preclinical background that is the result of decades of research and product development that we have conducted in very close collaboration with 5 academic institutions in Sweden and Japan. The rest of our founders, they are, I dare to say, one of Sweden's leading teams in immunology, and together with the rest of our colleagues, we comprise a highly qualified R&D team who specializes in different parts of preclinical and clinical studies. Now, what we do as an organization is that we are building iCoat towards commercialization of our products that will target the large and non-cyclical organ transplant market. And this is something we do based upon our IP-protected platform technology centered around our research, upon which multiple product candidates are emerging, all of which will have a so-called orphan drug designation. Our lead indication and lead product is called TUM12, which will enter its first in-human study later this fall for kidneys. But in parallel, we will continue our preclinical research in collaboration with the universities from where we come from. This gives us access to world-class research, great colleagues, and effectively keep our operating spends at a very low level. So iCoat is an effective and cost-conscious firm that is working towards clinical trial and subsequent commercialization. We expect that in 2025, our first product and its first indication will be ready for commercial launch, upon which our prognosis indicate around 200 to 220,000 transplantations taking place around the world. Now, our first indication is for kidneys, but our ambition is to also move forward with heart and liver, and effectively we will then cover the absolute majority, more than 90% of these transplantations. This product thereby effectively have a market potential that has been estimated to around $4 billion. And as of now, there are no direct competitors to our product TUM12. If we move to the next section. Our team constitutes the 5 founders who have been working for more than 20 years together now, and myself, and those are Professor Bo Nilsson, Professor Alireza Biglarnia, Professor Marianne Jensen-Wern, Professor Joji Teramura, and Professor Kristina Nilsson. Together with David, we're also joined in our management team by Christer Sävholm, our head of toxicology. Christer has previously held similar position in various industrial and large pharma firms, including AstraZeneca. And finally, we're also joined by Inga Dahlfeldt, our director for clinical trials. Inga has been successfully planning, leading, and executing similar trials to the one we are about to commence for the past 30 years. So taken together, we are a management team with different commercial and industrial and academic backgrounds. That's all taken together comprises a complete team, so to speak. If you go to the next page, please. And this is something that stands out, our statement that iCoat has a complete team. Most early stage pharma companies, they comprise a group who come from the same research background. We do not. We have different specialities, meaning that iCoat Medical as a company covers the complete research chain from biochemistry and basic and applied immunology through translational and preclinical work into clinical medicine and subsequent commercialization. So while being a small firm, we have no operational gaps, but rather we are fully set for the trial, which will commence in Q4 this year. We go to the next one, please. We're also very proud of the board of directors and our key opinion leaders who govern and support our development. The board is led by Hans Larsson, former chairman of the board for Svenska Handelsbanken, as well as one of Sweden's premier industrialists. We also have Bertil Villard, one of the founding members of Inga and former head of corporate finance at Alfred Berg on the board. Bo Nilsson and Marianne Jensen represents the core of our work, whereas the fifth member is Martin Åmark, who on a daily basis works as the CEO of Xbrain Biopharma. Are on the other spectrum of innovation when it comes to pharma products. They are building generic products, but they have been very successful in the various finance and partnership developments across the world. Also, we have 3 key opinion leaders that represent iCoat, one in each of our 3 main markets. Professor Steven Sachs in Europe, Professor Raya Kanaswamy in the United States, and Professor Katsunari Tanabe in Japan. So that is a short intro about iCoat. If we go to the next page. So what we do is that we address the great medical need that is the worldwide shortage of organs suitable for transplantations. It's not only just a shortage, but the waiting lists and people who have to be in dialysis as a fact of it, they continue to grow every year. Just to exemplify, less than 10% of the global need of organs for transplantation is being covered, and each year kidney disease kills more people than breast or prostate cancer. So this is a very big problem. Now, main reason to this deficit of organs is that during the transport from a donor to a recipient, any organ experiences a lack of oxygen during the transport called ischemia. Because of this, when the transplantation is conducted, a so-called ischemia-reperfusion injury occurs. That injury damages the organ and subsequently activates the recipient body's immune system. This ischemia-reperfusion injury is the center for our research and the area which we build our products upon. And what we aim to do is to significantly decrease the associated levels of organ damage that comes from this ischemia-reperfusion injury through our product TUM12. This will lead to not only an increased life length of the organ being transplanted, but it will also extend the pool of organs that are available for transplantations. So taken together, these 2 factors will help reduce the waiting list for transplants all over the world, something that is greatly needed. If we go to the next one, please. So here I just want to give you a brief intro to this research area that we focus on and what it implies. Our focus is ischemia-reperfusion injury. That is a so-called pathophysiological mechanism which leads to a thromboid inflammation and tissue injuries that occurs in many areas, not just transplantation. And to simplify it a bit, ischemia-reperfusion injury can be described as a 2-step process. First, a lack of oxygen leads to cell injuries and changes in the cell surface. For example, when a transplant is removed from the donor's body and transported. Thereafter, the recipient body, its immune system is activated because of this lack of oxygen, which leads to an attack of these altered cells that causes this so-called thromboid inflammation. Now, thromboid inflammation occurs not just in organs being transplanted, but in many of some of the most common causes of death in the world and the connected treatment of it, such as cardiac bypass, myocardial infarction, and stroke. So in theory, this is the very vast universe of indication for iCoat Medical, but we start out with solid organs for transplantation. And this is something we do through this product TUM12 that I will now cover a bit if we go to the next page. So the proof of concept and connected mode of action for TUM12, that is something we proved in 2012. And basically the underlying issue is this ischemia reperfusion injury, which always develops during a transplantation. It doesn't matter who the recipient is, what kind of organ being transplanted, or how well the procedure goes. The IR injury always happens. And what TUM12 does is that it is a so-called ex vivo treatment of an organ, meaning that it is treated outside of the body before the kidney, for example, is transplanted into the patient. And it acts as a coat, which forms a barrier and protects the endothelium and compensates for these damaged glycocalyx. So effectively, the mode of action has been confirmed, and it is confirmed through a significant protection against these kind of injuries. In the bottom, you see a very simplified illustration of this. To the left, you have a cell membrane that is attacked in a normal state. But in our case, when TUM12 is inserted, the product acts as a protecting coat, which creates an impermeable coating around it before it leaves the body through the urine. This is something we have demonstrated in numerous in vitro studies, as well as in 4 large in vivo studies on pigs. Pigs are very good to work on, and I will explain why, but first I'm just going to demonstrate how the product looks in more detail. So next one. TUM12 in essence comprises 2 main parts. It is a PEG and it is a so-called phospholipid. And what it does is that TUM12 interacts with the phospholipid of the cell membrane, shown here as what you see on the left side, part of a cell, and acts as this kind of shield. To the right, you see a microscopic picture of a cell that has been treated with the TUM12 and also a fluorescent fits to demonstrate this kind of coat. Very, very strong protection. If we go to the next one. The preclinical work we have done has been largely centered on studies on pigs, and they are very suitable for large animal models for kidney transplantations in particular. And this is because the pig's kidney is very similar to the human organ with respect to both anatomy and physiology. And in our case, the transplantations we have conducted, they have also been performed in the same manner as are done in the clinical setting for human patients. So what we have seen is that TUM12 demonstrates very strong efficacy data, that it shows an improved kidney function and reduced inflammation rates. So if we go to the next one. This is a very simplified but effective way to show how we've built our studies. And basically, donor donates both its kidneys to 2 piglets, that is pig siblings. One of the kidneys is being treated with TUM12 and the other one with a control product, effectively excluding all external factors and just demonstrating the results of TUM12. Next page. And the results are truly very astonishing and very promising that it will translate well into humans. This is a bit complicated one, but what you see on the left side of this page is creatinine levels measured in the control group and the group treated with TUM12. And here we see a statistically proven improved organ function for those kidneys that have been treated with TUM12. We also in the middle see a lower reaction and activation level of the innate immune system, showing also, which is also demonstrated to the right, where we have presented various forms of common biomarkers. These ones demonstrate different forms of systemic inflammation. All of them are almost non-existent in the case of TUM12. So this is very promising for kidney transplantation, but It is based upon our work on ischemia reperfusion injury as a general concept, and thus we see great potential for a full pipeline of indications. So if we take the next one. This is the pipeline we're building. It is centered around our patented work, and we are developing two product categories, TUM 12, which is for cold ischemia, and TUM 20, its successor for warm ischemia. Each of them come with three main indications, but we also see future preclinical projects emerging around our work. However, as we're working with ABE now, we are financing the first clinical trial, which will be for the indication of kidneys. So if we go to the next page, I want to show you where we are right now. And that is here in May, where we're raising about 40 to 45 million together with ABG. The book is filling up quite fast, but there's still room there. We will then file our application in early July after the final preparatory and administrative and financing aspects have been completed, before we commence the trial in early Q4 this year. In parallel, we are also progressing with future indications of TUM 12, as well as the second product group, TUM 20. This work is already started in April this year and is currently continuing with early positive outcome for warm ischemia as well. So taken together, there are very strong and significant value creation steps that will happen over the next 18 months before we expect to IPO in late 2022, which we will do with both safety and efficacy data to support our work. So if we go to the next page, please. I want to conclude this short intro by stating that iCoat Medical is something truly great in our opinion. We are developing novel treatments for organ transplantations and several other very large indications. There is a massive market we're heading towards in a couple of years, and there is no known direct competitor. We have all organizational aspects in place, and we are progressing according to plan and looking forward to the IPO in about 18 months. With that, I want to thank you, and me and David will answer any and all questions you may have. Thank you.
Great, thank you very much for that presentation, Peder. A few questions. Firstly, I was wondering if you could elaborate on how the treatment is administered practically. You said it's an ex vivo treatment of the organ, but practically, how do you go about treating the organs, and how feasible is that method of administration in current workflows? Does it require any kind of changes to how the transplantations are carried out today, or does it kind of flow in easily?
You worked in the area before with this kind of product, David. Do you want to take it, or shall I?
Yeah, the practical aspects of administering this product is very straightforward. Whenever you procure an organ, regardless of whether it's a live or deceased donor, you always perfuse the organ with a crystalline type of solution. And this is simply an additive to that solution. It's compatible with whatever solution you use that is on the market. And on top of that, it is also compatible with what is called machine perfusion, which in turn can be done both hypothermically and normothermically. So the simple addition of this in the standard of care perfusion solution is what takes place.
Okay, great. And from a regulatory point of view, this is to be considered a drug as any other, despite being, given on or administered to an ex vivo organ, as it were.
Yes, the regulatory distinction between different product categories are very interesting, and this is a very very broad and deep discussion. But this is something that we have looked into in great detail and have discussed with the Medical Products Agency in Sweden, and are comfortable with the pharmaceutical designation that we're going with.
Great. And looking ahead to the clinical trial, the first clinical trial that you're planning for at the end of this year, I was wondering if you could comment on your CMC situation. I mean, primarily the manufacturing side. Do you have enough product for the trial, and how's that being produced and so on?
Yes, we do. We are collaborating with a prominent GMP certified firm called Basic Pharma, based out of the Netherlands. They are currently preparing for the tech batch release, which will happen in early July. And we have been working closely with them for quite some time, and they have also been fully vetted in terms of capability and certifications, and they have passed it with flying colors. So there were some early delays in the past for us with Basic Pharma due to a global shortage of vials, as they all went to the COVID-19 vaccination development across the world. But as of now, and with the current presented timeline, everything is progressing according to plan.
Okay. And a question on the upcoming trial. What can you say in terms of the setup of that study, endpoints, size, and so on?
You want to take it, David?
Yeah, so this is, of course, a small initial trial whenever you do a first-in-human trial. I think the important part to focus on in this initial trial is that we're going to be able to collect a number of really important biomarkers in the trial. So when you have the ischemic event and subsequent reperfusion, perioperatively, you can collect blood samples just very similar to what you saw in these pig models, directly coming out of the transplanted organ. In essence, you take a sample from the renal vein in this case. And this can, with a very high precision, show to what extent we have been able to minimize the ischemia reperfusion events that happen. And that is something that we can accomplish in a small trial size with just around 10 patients, or a little over 10 patients is going to be possible to attain that. And that's then going to form what we believe a very effective base for an adaptive phase 2/3 design, which we believe will form the basis for registration on the market.
Okay, great. And a question on the TUM20. On a high level, how does that differ from TUM12 in terms of the kind of the mechanism of action and then the design of the molecules?
Yeah. So the ingenuity about this whole concept is really the effective coating that was outlined in the presentation. And this is really unique. If you want to try to accomplish the same type of effect by administering specific inhibitors, you would have to give something like 10, 15, 20 different antibodies and inhibitors of various pathways and cytokines, which in practice is absolutely impossible. So the basic inert coating that TUM12 provides is absolutely essential. On top of that, in certain ischemic situations, particularly with myocardial infarction and stroke, some of these pathways are going to be further augmented. So the TUM20 is a further addition of specific inhibitors for certain pathways. It could be, for instance, heparin, it can be specific complement inhibitors, and the one that we're driving forward right now is one that targets the coagulation cascade.
Okay, interesting. And obviously, for those indications, it wouldn't be the treatment of an ex vivo organ. It would be treating a live patient. Out of interest, what would that administration method look like? Is that likely to be an IV injection, or how do you envisage that?
Yes, so that's where the preclinical work is focused on optimizing the approach for maximizing the eventual clinical utility of those indications. The good thing is that there are a number of interventional procedures that are done routinely clinically today for a variety of ischemic events. So if you have a myocardial infarction, for instance, a blockage in a coronary artery, you can do an endovascular endoplasty and expand that blockage. That creates an ideal opportunity to also inject a treatment, for instance, TUM20. The same goes for a fraction of the stroke patients that do mechanical thrombectomy, and that also creates an ideal situation for administering. Now, the timing for giving this type of treatment systemically needs to be assessed more thoroughly before we know if that is a viable option or not in a more comprehensive situation where you don't need this type of targeted delivery. And the advantage is that the toxicological profile of this substance is absolutely pristine, which means that we can load a patient with very high doses without any side effects. We can't rule out that a systemic administration is relevant as well.
Okay, very interesting. Thank you so much for your presentation.
Thank you for having us.